at Madison will be involved in combining machine learning, deep learning based techniques with experimental chemistry to solve interesting problems in biology. So with that, I'd like to welcome Anna.
Thank you so much. So thank you so much for the invite. This seems like a really fantastic group, and it's a real honor for me to kick this off with talking about a topic that I care and love a lot, which is AlphaFold and its friends, because it's really one model out of many that are really changing the game of what we can do for predictive structural molecular biology. And so when I think about AlphaFold and I want to think about how to understand it, for me, it actually goes back to Yehoshua Bar Hillel, as it may for some of you. So Yehoshua Bar Hillel was a philosopher, mathematician, linguist at MIT who passed away a number of decades ago. And among other things, he thought and philosophized about this problem of machine translation. Let's see if I clear some messages here. Oops, where'd it go? Okay, so machine translation is this task of, given a sentence, can I translate it into another language? Something we really take for granted nowadays. But research in the field of machine translation really picked up around the time of the Cold War. There was a lot of interest in translating Russian to English and vice versa. And so our friend Yehoshua Bar Hillel at one point wrote a treatise, a demonstration of the non-feasibility of fully automatic high-quality translation. So there's this influential essay in 1960. He laid out that this he thought would never be possible. And so I just want to take some quotes from this. So here, inventing a method for fully automatic high-quality translation is just a dream which will not come true in the foreseeable future. And he said the reason for this, it is an old prejudice, but nevertheless a prejudice that taking into consideration a sufficiently large linguistic environment as such will suffice to reduce the semantical ambiguity of a given word. That's a lot of words, but if I sum that all up, the hard thing about translation is context. And we all understand this about language, that the meaning of a word depends on the context that it's in. And to me, when I read this treatise, it was really interesting because nowadays we have apps on our phones that I can just go to Sweden and point it at a menu in Swedish, and it immediately translates it for me. So I don't mean to hate on Yehoshua or anything, by no means. I just think it's so interesting, and it's really hard for us to predict what can and can't happen. And so interestingly, so this essay that he wrote about how there's no way for a machine to really nail down context of words. This and other people. Other writings and philosophizings led to influencing a government report, which was known as the ALPAC report, in which they conclude there's no immediate or predictable prospect of useful machine translation. And that government report, then historians say, led to what they call the first AI winter. So we fast forward. In 2020, there's this new algorithm, a couple AI summers, AI winters, that many of you are familiar with, which is this AlphaFold algorithm from DeepMind. And so this, I recall being a grad student on Zooms at the time, and it completely changed what people thought was possible for single structure prediction for proteins. And so this I'm just taking from a blog post. But so there was this crowdsourced competition that happened every other year in CASP, where experimentalists would gather data from X-ray crystallography or other methods. They would blindly hold out that data somewhere. Teams creating various structure prediction tools would then make a blind prediction. And then the team of assessors would compare the experimental model and the computational model. And so AlphaFold completely blew away. So there's now these sorts of images are almost just routine. We can see the comparison of AlphaFold models with these single structure models. So it's really cool. So, okay, so things I want to talk about today. I want to dig in a little bit more to what is AlphaFold? What's the architecture? What's it come from? But AlphaFold, I view it as really a one of things that are more broadly considered as protein language models. And so so getting back to that question of language and context, that's to me what's really interesting. And I want to try to tell you by the end of today that those architectures are actually theoretically pretty well suited to consider protein structure. And there's even some equations, and depending on your interests, you can dig into them and we can chat more. You can ignore the equations, but so that's sort of things I'd like to be able to tell you about today. And along the way, please, of course, if people are coming from very different backgrounds, feel free to stop or ask questions or say, yeah, that didn't make any sense. Please say that again. Okay, and just so another semantics thing, we hear a lot of words about deep learning, AI, machine learning. To me, I have this maybe sort of hot take that that AlphaFold is not AI. So to me, AlphaFold is predicting something that's really cool, but take a expert computational structural biologist, give them a sequence, and odds are, unless it's something they've seen before, they never in 100 to 200 years would be able to tell you what it might fold to. So I view AlphaFold as in the scope of very good and useful deep learning. But I think artificial intelligence, there's whole other areas of other areas of study, including theoretical cognitive science that deal with this. But maybe I shouldn't tell people I'm writing rants to that. Okay, and so then also just wanting to lay out some semantics. I'm not sure everybody's background here. So writ large, there's sort of two flavors of machine learning, unsupervised learning and supervised learning, so to give some context for these models. So unsupervised learning, if you've ever done PCA, you've done unsupervised learning. So unsupervised learning is when you're given some set of data that is unlabeled, and broadly speaking, you have some sort of model that itself is trying to find patterns in the data. And so PCA or TSNE is an example that a lot of people have done of that. Supervised learning is where, the classic, I have pictures of cats and dogs and I want to try to come up with a classifier to tell me what is what. So the data is assigned labels, and in the same way, so AlphaFold is an example of supervised learning in that I have sequences and then I have outputs, which are essentially the task that I'm asking it to do. But then also, of course, lines currently are being blurred. AI companion is on. Cool. So unsupervised learning, if you have a model without labels and you might find out ways to, after it's learned a distribution, to then sample from that distribution. And so that's then where aspect models related to generative modeling come from. So DALL-E is pretty good at that, I hear. So there's things in the protein space as well, models like RF diffusion and chroma, where they've essentially been given a distribution of structures and are now also able to sample and sample from that space. And so that's pretty cool. That requires both some unsupervised and supervised learning. So, okay, so the fundamental reason why AlphaFold and all these friends of AlphaFold work can be, to me, summarized in just this figure here, which is coming from this 2011 paper. So protein sequences have to hold information about their structure, because that's sort of all they have. And so if you look across evolution, and so people look across evolution and they say, oh, here's a hemoglobin from fish, and here's a hemoglobin from humans, and here's a hemoglobin from cats. And they all look pretty similar, but one can realize that there are positions where these residues vary together. So evolution, random drift is pushing things one way or another. Some of these positions co-vary. And the neat thing that people figured out was that some of those positions that co-vary, it's because they're close in 3D space. And so this should make a lot of sense, because if a mutation happens here, but it's important for the function of the protein that it's hydrogen bonding with this residue over here, then if I mutate one, if I need to preserve that hydrogen bond, I better mutate the other. So that's sort of that's the essential reason why. And if you look at this is really just a 2D array. And so, okay, so here's the figure of the AlphaFold architecture, which if you read this paper, you have probably seen. So the way that AlphaFold is set up, so you have this input sequence. It goes and so outside of AlphaFold, run some sort of sequence similarity search, so BLAST or something like that, that you might have done yourself for some sort of project, it generates this multiple sequence alignment, or MSA. So for a given protein, depending on how many homologs there are, this might be somewhere from a couple hundred to tens of thousands of proteins. And so then also, if you're running AlphaFold, if you depending on certain options that you can turn on, you might also then do a structure database search. There's also this option to put in so-called templates, which are 3D coordinates then of other related proteins. And so the representations then that AlphaFold creates and then uses, there are two main ones. So one of these is called the MSA representation. So again, this is MSA, really, you can think of it as this 2D object where one of the axes is the variance from across biology that across evolution that we found, and then the other axis is the length of the sequence. Both of these get fed into sort of what's the first stage of AlphaFold, this so-called Evo former. And this is essentially a stack of transformers, which we'll talk about more later. So the transformers and Evo former do their thing. They come out again, and then they go into the so-called structure module. And then the final result of this is the 3D structure that AlphaFold predicts. And the classic set of options that is sort of the default options is then this structure gets used as a template and fed back in and is done multiple times. So this so-called recycling. So for those who actually have been following DeepSeek or other things, there's now the buzz is a chain of thought. So sort of, it turns out you can make something like ChatGPT better by getting it to sort of think through and think multiple times about what it's doing. And so some people in a very simple sense liken AlphaFold recycling to that as well. And it really sort of, I mean, it's a sort of really simple thing, but it's sort of one hypothesis that had at one stage, and then that serves as a starting point in that process of recycling. And work that I view as being very foundational for the field was this paper. So Sergei Ovchinnikov, who now has a group at MIT, was the senior author on this. It appeared in physics review letters, so maybe it has something to do with physics. And they had a hypothesis about this AlphaFold architecture, which was basically that they thought that it might be that this MSA, this first step, was sort of providing an initial guess for AlphaFold, and that the structure module is then sort of using that guess, starting from sort of just rough contacts of where it might be, to then refine and then give back a more cemented idea of the 3D structure. So to test that hypothesis about how AlphaFold works, what they did was they took a longstanding problem from the field of Rosetta. So Rosetta from the Baker lab had a long-standing task was to say, if I have say I have like 1,000 models of a particular protein, and I want to know which of those is closest to what might be my actual experimental data or my native state. So that problem, they call it decoy ranking. So the multiple models are often referred to as decoys. And so this paper, they found that actually the structure module of AlphaFold was better at ranking decoys than the current best Rosetta methods at the time. And so this view now really shapes how a lot of people in this field think about how AlphaFold works. So sort of one step, this evolutionary information is giving this sort of two-dimensional positioning of what should be contacting what. And then this structure module is taking that, going from 2 dimensions to 3 dimensions. And so people have hacked this a lot, and that's been neat because it's open sourced. And so one fun hack, so the original AlphaFold couldn't do cyclic proteins, but people realized if you change the positional embedding, so instead of just going from 1 to L and then ending it at that, then if you add something on after L to go back to -1, that they could then get it to predict things in chains, and showed that it did very well at predicting things in circles as well. People have shown, so again, the original AlphaFold 2, it wasn't trained as a ligand binder, but folks have shown that if they take that pair representation of AlphaFold and sort of consider it, can you train something on top of that to predict ligand bindings? The way that these authors set this up is they said, what if we sort of give AlphaFold a template, a sequence, and then these so-called bait amino acids, which is sort of fun. So they're sort of hacking it, treating each of these as a different, another chain, and then asking, do any of these quote bait amino acids bind somewhere on this backbone template? They found that probability of binding is competitive with other blind docking methods. And then something that I've been involved in working on, and that there's now multiple ways to do now, is to perturb that initial sequence input into AlphaFold. So a couple ways in which people have shown that. So the first way that people showed this was through what they call shallow MSAs. So if my protein has thousands of homologs, but I say I want to add a lot of noise to that. So instead of the 1,000, I'm going to just randomly sample 100 of those. And if I do that repeatedly, so I'm basically just adding noise on top of whatever coevolutionary signal is there. That is a way to get AlphaFold to generate multiple conformations. Another way is to just simply ablate certain residues at certain positions. People have found that can cause AlphaFold to switch predictions back and forth. The hack that I made was I showed that for certain families of fold switching proteins, that there are families where there's actually pockets of evolutionary signal for one spot, for one fold or the other fold. And so if we developed a clustering method that teased out those different sets. And so then that different MSA level information gets put into AlphaFold and is able to predict different conformations. I won't talk a ton about that more, but always happy to chat more about that after the fact. So all this works because protein sequences hold information about structure. And so here I have this cartoon of this idea of so I have these sequences and they go through my supervised learning algorithm of AlphaFold and they predict a structure. But post AlphaFold, concurrently with AlphaFold, there's been this explosion of so-called protein language models. And to me and the rest of the computer science world, they're really cool. And I want to tell you why we all think that they're really cool. So it's because they use unsupervised learning. So instead of the task being, I'm giving you the structure that I want you to predict, the task is, I am going to hide some of these residues from across all the domains of life. And then a flagship model that is studied a lot is called ESM, stands for evolutionary scale modeling. Actually, just curious, can I get a show of hands, like how many people have heard of ESM? Okay, cool. I know you can hear it. And okay, cool. So ESM comes from the esteemed biochemistry research halls of Facebook AI research. And it's a model that gets analyzed a lot. So this task comes directly from the same types of things that created ChatGPT. And the task is often referred to as masked language model. And so it's this idea that you're hiding residues, and then the model's job is just basically to memorize what residues should have been there. Essentially, what makes for a protein that is now evolved and present in our sequencing databases. And then what does one do with a model like this? What people have then shown is that if they then take sort of the fattest, thickest parts out of ESM, the so-called embeddings, this multidimensional space, this latent space of ESM, they've shown that they can then take those embeddings and do some quick training on top of that for some sort of supervised task that they care about and predict all sorts of really neat things. So the structure prediction version of ESM is called ESM-Fold. It rivals AlphaFold in accuracy, even though it was never trained on those multiple sequence alignments. It was simply trained on a whole bunch of sequences and then fine-tuned a little bit at the end with structure information. So another task, function classification. What type of enzyme is this protein that I have? Is it an enzyme? Language models have been able to do that state of the art well. Another interesting thing as well, people have been looking at language models more broadly in the context of entire genomes and find that they can train models based on them that it has predictive power for which proteins are co-regulating each other, so which proteins are appearing in operons together. So it's really still an open question. What information can be just sort of purely encoded in sequence at that level? And so something too that is just sort of striking, I think, to show side by side. So this is a figure from a NERV's paper. So this is the same same architecture, but just different point of view that we were looking at before. So this is the entirety of AlphaFold. So sequence to pair represent. Oops, go back to pair representation. MSA evoformer pair MSA structure module. If we look inside the evoformer, this is opening the box of the evoformer. There's more attention. I want to tell you more about what exactly is self-attention in these transformers. Why does this work? But what's really interesting is if you compare all these steps in this architecture to what is going on in ESM, there's just one attention operation, essentially. But yet, all that just training on more sequences. And so if we think about the amount of data, say, that AlphaFold versus ESM have to train on, AlphaFold, currently in the TDB, there's maybe 200,000 proteins for which we have sequence and a structure. So that is maybe not that much data in the grand scheme of things. But we have, I believe the number is 137, gosh, I can never get this right if it's million or billion, sequences, like proteins ever sequenced. I think it's 107 billion, right? Yeah. Billion. I think it's billion, which is many orders of magnitude more than 200,000. Okay, and so to understand sort of why such a simple self-attention, why should such a simple self-attention thing be able to give us all this predictive power over proteins? And so going back to machine translation, and did Yehoshua's predictions bear out or not? So in 2016, the state of the art in machine translation were so-called recurrent neural nets. And so some common recurrent neural nets include gated recurrent units, LSTM, so long short-term memory. I don't work with these. I don't know much about them, but the essential of them is that in doing natural language processing, translation, even now if you're doing protein language models, people talk about tokens. So token is sort of like the basic unit of whatever is being fed into the model. And so in this case, so these recurrent models consider a sentence one token at a time. And so if this is the first word of my sentence, this gets fed in, the RNN then continues to accept tokens, and then along the way, it's outputting what it thinks the translation should be. So that was state of the art in 2016. In 2017 was this seminal paper titled "Attention is All You Need," a title that has now been parodied a lot. And so this really was a breakthrough for the field. Funds sociology. So this is written by 8 authors, and they are all joint first authors, and they decided author order by a random number generator. So transformers, which are the architecture that uses this so-called self-attention mechanism, take in the whole sentence at once and then do learn context, which is the thing that seems to be hard for language. And then there's multiple ways to sample. You can either sample one token at a time, or you can return an entire sentence at once. And the neat thing then is that transformers allowed there to be this breakthrough in natural language processing because it basically changed the dimensions of the problem. So previously with a recurrent neural net, if you had a sentence of length n, basically you needed to do n operations. But a transformer then took that and turned it into basically just one matrix multiplication problem of taking in all these tokens, doing essentially one matrix multiplication, which is the transformer architecture, and then having this model to then sample from. And so this attention mechanism, so here's a fun example of the thing that made Yehoshua nervous about language. So if I read this sentence, The animal didn't cross the street because it was too tired. The word it, I know because I have a brain that this refers to animal. But if I read the sentence, the animal didn't cross the street because it was too wide, it is referring to the street, probably. Um, uh, and so the cool thing then is in this, with this attention mechanism, one can then go and there'll be a couple of vectors involved. So one of them is called the query vector, and so the query vector is essentially at each point attending to different words. And so the query vector for it in this trained language model, on this sentence, it's looking at animal. Here, this query vector for it is looking at street. And that, this matrix multiplication, I mean, it seems simple and it's easy to say after the fact, but this sort of solves the context problem of language. And so the attention mechanism is a really great thing to know if this is, possibly this is all old news to you, but it essentially takes a vector and then performs an operation to turn that vector into three things, which are the query, key, and value. And so people often call this embedding. So if I have this sentence, which is just 2 words, so my sentence is thinking box, I'll embed it. And I just have these matrices that it gets multiplied by. And then I have this K, Q, and V, these matrices now. And so these are things then that as I would be training a language model, these are matrices that are being learned by the model. And then all that gets done in the self-attention mechanism is I just multiply sort of my original sentence as viewed as a query, and then my sentence as viewed as a key. And so these names, one can really sort of intuitively think of them in terms of YouTube. So like here, my query might be these 2 words of swimming rabbit. And then the key that my model might be then trying to learn is basically all the keys, across all of YouTube. And so there's a lot of keys out there, some of which have to do with swimming rabbits, but there's many videos out there that don't have to do with swimming rabbits. And so by then doing this matrix multiplication, I'm basically trying to find which of the keys match best with my query words. And there's a value matrix as well that is sort of telling me for any given match between my query and key, how much should I pay attention to that? And so that's all to say that it turns out that these transformers, so that really simple, just matrix multiplication of these 2 things, they are actually structured very similarly to old school POTS models that people developed starting 30 years ago for trying to model this problem of which residues covary together. And to me, this is a really interesting thing because I think there's many people in biochemistry in particular and other areas of science who, are appropriately leery of deep learning because sometimes it can feel like a black box that we don't know how to prod or understand. But to me, this is a result that I think is really neat because there is, it turns out, this sort of strong underlying physics-based reason for why these transformers are good at modeling proteins. And so this is just this little paper in this Pacific Symposium on Biocomputing, which I learned later the authors submitted there because it meant they might get a trip to Hawaii to submit it there. But I really encourage you, if this is interesting to you, to take a read of that paper because they'll do a better job of laying it out, in many pages of text than I will in a couple of slides. But so essentially here I'm showing, this is a contact map. So if I'm thinking about in protein space, there are these 2 axes that I can consider, both if my protein is length L, I have these 2 axes of length L. And then so the old school version of this, the problem was then how do I learn from a family of proteins which of these positions are covarying? And then essentially I would hope to use that information to predict a structure. And so in that old school POTS model formulation, what people found to be surprisingly effective. And so by people, so folks who've worked on this, so my mentor when I was at Google, so Lucy Caldwell, Debbie Marks, who's at Harvard, so Sergey Ovchinnikov is someone, there are a number of other folks though who have also sort of come to this sort of conclusion. So basically they found that if we consider this type of potential, so if I just consider basically pairwise couplings and then some positional bias per position, they found that that's actually a pretty fine, sufficient model to be able to then say, maybe I want to learn a distribution of, say, like all the sequences that might appear in the family of hemoglobin. And then if I want to draw from that, produce something that is a new hemoglobin, that super simple model actually does a pretty decent job at doing that. And so this matrix here, I can think of this as if my protein is size L, I can think of this as an L by L by 20 by 20 matrix or tensor in this case. And so essentially what that old school Potts model then is learning is it's learning for every 2 positions, I want to learn basically the likelihood of if there's a cysteine at this position and a lysine at that position, how often do I observe those 2 appearing together for this protein family in this position? So, okay, so L times 20 times 20. So if I have 100 residues, that's about 2 million numbers that I have to learn. That might still be really useful, if this is a family that I care a lot about, but that's still a lot of numbers, one can imagine. For all, one would like to be able to figure out ways to do this more broadly and generalizably across multiple families. And so if one goes back then also, so in this context, there's been a lot of work spent on how to use other methods such as message passing, mean field approximation, pseudo likelihood methods to just given basically a finite number of sequences, how to estimate that 4-dimensional tensor. And this is the field then that I view sort of as AlphaFold as emerging from, is this line of work. Okay, so now I want to convince you that we can rewrite that Potts model as a transformer, and we'll get useful things by doing that. So out of that model, I'm going to consider just a term basically for one position here. So I'm going to call this, I don't know, position 13 and position 20. And I'm going to consider that there can be any of 20 amino acids at that spot. So that's a 20 by 20 matrix. And now I'm going to say, instead of like learning a new one for each set of positions, I'm just going to assume that I have some pool of shared feature matrices. Like I could probably believe that there's going to be some set of features that determine if something's in a helix and is hydrogen bonding with something else. And I can probably then assume that might be different than if one of those is in a loop and the other is in like a disordered strand somewhere else. So hopefully that sort of seems reasonable, that there should be some shared commonalities across these. And so I'm going to say that I have 8 of these feature matrices. And then all of a sudden, basically, I am back at having an attention mechanism. So this H is what gets commonly referred to in a transformer architecture as the number of heads in your transformer architecture. And my WV here, this is this 20 by 20 matrix. I'm just going to basically rewrite this a little bit. So I'm going to basically say, instead of having a 20 by 20 matrix, I'm just going to factor it one more time to just basically have something in terms of for at each position. I'm going to say, I think that it's probably the case that if something is here versus there, that they should be similar. And so then so people refer to this formulation as factored attention. And so I've written this in a very telling way because there's a Q and a K and a V. But basically, we're now back at a transformer. And all it took was just basically saying, I have a Potts model, but I'm going to assume that I can share features across those different positions. And the neat thing then is that if I have that model, my parameter count should go way down because I could now apply this. I don't have to go and relearn a new POTS model for every protein family that I want to study. And so if I just compare, so these are the two expressions. If I have a POTS model, for how many parameters would there be versus if I have one of these factored attention models. So in this factored attention, I would have the number of heads times L times then some D, which is basically the one parameter that I would be controlling, which is sort of the depth that I want to allow my model to have. And so then if I just look at the length, if this is the length of a protein versus how many millions of parameters, so the POTS model is going up quadratically, but the others, it's more log or linear in terms of how many parameters it has. So that's nice, just because I can write it that way doesn't actually tell me that's how those protein language models might actually be working. So a really useful study that they did in this paper is they then said, okay, what if we make one of these factored attention models, and I'm going to change the number of heads. I'm going to compare this to a full POTS model. And what they found is that the precision goes up to the point where that when they were at about 256 or 512 different heads, so sort of different feature sets, that was about equivalent to basically relearning a new POTS model for every single protein family that they studied. So another finding that they found here, but is common across all of these models, with protein language models or AlphaFold, the accuracy goes up as your MSA gets deeper. This hopefully should make sense, because all of these models, basically, if there's more sequences out there, there's more to learn from. So okay, I have a little research story to talk about at the end, but I sort of want to pause there. So to me, this is like super cool, but I don't know if it's sort of like completely lost everyone, but my takeaway from this is that black boxes, they seem like things that are black boxes, but there are physics and statistics based ways to link these two things to interpretable models that we can understand. And there are people doing this sort of research. And really, it's these advancements that then push people to figure out better architectures for how to model all of this.
What I think.
Yeah, that's a good question. So you're asking about sort of the pairwise things versus the positional encoding. And I would need to think on that. Yeah, that's a really good question. I bet it is discussed in this paper, but yeah. So for context, so a positional encoding is something in a transformer that is added to basically after doing that matrix multiplication to sort of give the model back information about where in the sequence or in the sentence that the model is looking at. So one can imagine there might be different effects close to the start of a sentence versus the end of a sentence. And so it's a very fundamental part of doing that matrix multiplication to get out context again. All right, I'll continue chatting. So building on that former work, something that I've been interested in and others have been interested in is protein isoforms. So because we viewed them as a potential pitfall for evolution-based structure prediction. And I'll try to say what I mean by that. So this is a structure of an enzyme. And through biology, through alternative splicing or other means, it can be the case that proteins get expressed from a gene, but some snippet of the protein, so not the entire protein. And so some of those isoforms are functional. Many aren't. Many then if in disease or other contexts things get dysregulated, sometimes isoforms, mistranscribed isoforms, translated isoforms can cause a problem. But so something sort of interesting to think about an isoform is that, so out of this enzyme, there is an isoform that exists out there that is basically just this chunk of it. And so if I color where that isoform falls on this structure, it makes this, sort of twisting and turning around because it's only part of that full length protein, of the structure the full length protein forms. But if you put it in AlphaFold 2, AlphaFold 2 will tell you with really high confidence that this isoform folds to exactly that shape, which if you were to go and then try to actually do a structure experiment on that isoform, one would quickly realize it would not be holding this shape. There's nothing to be like propping it in that position. So AlphaFold thinks is very high confidence that that's what it should be. And so it sort of makes sense why that might be the case, because AlphaFold learned over a bunch of MSAs of full length proteins that there were these contacts and this and that. And but so we were curious if those protein language models might fall prey to this same type of pitfall, because they haven't been trained on MSAs. They've been trained in this other way of this masked language modeling. And there was a period of time where there was a lot of hype that these MSA-free methods, have changed the game. They're better than AlphaFold. But so we did the study. We found that a bunch of these methods gave back the same answer for these isoforms that AlphaFold gave back. So OmegaFold was another model that garnered a lot of interest because it was trained in a similar way to AlphaFold, but only took in single sequences rather than entire multiple sequence alignments. But it had the same problem. There's really actually only one method that gave a different answer, and that was sort of interesting. And here's another example. So here's this full length protein. There's an isoform that exists that has this beta strand spliced out of it, and there's basically no physical reason now why this should fold to the shape it does, but AlphaFold and OmegaFold, ESM-Fold as well, gave back that it should still adopt that same shape even with the beta strand spliced out of it. And so we were digging deeper, and we realized we sort of wanted to see if we could generalize that finding. And so we did this study where we took predictions and put them in ESM-Fold, but basically the same thing. And we then sort of tried to chop away parts of the protein to basically see how much of the protein does ESM actually need to predict the contacts that it would predict. And the answer turned out to be not that much. The answer turned to be on average maybe like 20 to 40 residues in total, like surrounding that set of contacts. And so this is a visualization that Sergey made. And so here basically I'm showing, so everything outside of this green part is getting masked. And so what I want you to pay attention to is, actually replay it, is how long this set of contacts persists, even though the rest of the protein is being chopped away. And I'll just do it one more time because I like looking at this. I think I have another example too. So now here's a set of pairwise contacts that are pretty far apart from each other in sequence space. And we're going to mask everything before and after and between those. And they really stay a long time. And so our theory now about how these models might be working is that they've sort of maybe learned a set of those pairwise patterns of which residues are interacting. And this experiment demonstrates that by saying we can hide a lot of the rest of the protein so that it just needs a little bit of context. And so then it's just sort of drawing from this bag of different types of interactions that it's learned. And so in the conclusion of this paper, we did this sort of back of the envelope calculation where, so we were saying, so in UniProt, there's about 20,000 protein families. And if we assume they have an average length of 256, if we wanted to learn all of those sets of pairwise contacts, that 20 squared matrix times all the different, all the positions, in total, that would be about 261 billion parameters that one of these models would need. But if we assume that each position makes maybe at most 4 contacts, we said we were reasoning sort of if there's 2 neighbors in sequence and then maybe there's 2 longer range contacts, that reduces to about 4 billion parameters. And this is speculation, but it was interesting to us. That's sort of, so ESM2, so something that is common for language models is people will train the same model with different sizes and different numbers of millions or billions of parameters. And so the ESM2 models really start to taper off in their improvement after about 3 billion parameters. And so we suggested that a model that is segmenting a protein into common motifs, as our work suggests these PLMs are doing, offers a route to how they might compress those statistics. So this, I think, made a splash because, again, protein language models were something people sort of viewed as a black box of sorts, but through this, what was sort of a really empirical study, we show that we could consider them to just sort of have to be this model that has done a really great job, but of doing it. But essentially, it's basically just learned a set of interacting motifs, and there are doubtless other types of motifs and patterns that are beyond just pairwise interactions, but it has also learned those pairwise interactions too. So I think that's the end of what I wanted to say. So again, if you're taking nothing else away from today, what I want to tell you is that there's really a strong theoretical basis for why the architectures that underlie these protein deep learning models, why they work. And that comes from, because it turns out really nature is low rank and you can do matrix multiplications and get useful things. And so again, the reason for this, and so, the challenge of language, but also, again, the challenge of proteins is about how you learn that context. How do you decompose context and consider it usefully? And also, again, a takeaway is that we can and should work on interpreting these. Really, there's good things to be said about sitting down and spending quality time with one's neural net. So I think I will stop there, and super happy to take further questions or discuss more things. So thanks so much for listening. So these methods are not ones that are intended to give predictions of isoforms. So the way we did this study was taking known isoforms from UniProt or elsewhere. So yeah. Well, so I don't think there were ever models really intended to be used for predicting isoforms, though maybe I'm misunderstanding your question. But yeah, absolutely. Like if you have something that's an isoform and you put it in one of these models, it will give you back something that might look folded when it's really not. Yeah. So this was something, because people have gone and made databases of AlphaFold predictions of isoforms. Yeah. So I mean, the context in which people would use AlphaFold is where they know what the start and end position that they believe should be.
Along the same lines of trying to interpret these neural networks, I wonder if you get a thought up to rate the confidence of these models or get any sort of assessment of just the models.
Yeah, so I mean, you're probably aware that there is this PLDDT metric, right? That is part of AlphaFold. You're not? Okay, cool. Well, so I think AlphaFold is actually a really great example in deep learning of people creating a confidence metric for it. And so what I'm showing here is so it's the same structure, but it's colored by this number, this PLDDT. And so dark blue means it's more confident, and green means it's less confident. So in many contexts, it's useful. Here, clearly, it's not being accurate. But so the rationale why the authors of AlphaFold built in this confidence metric is sort of the community that they built it in, which was this structure prediction competition. And so they themselves didn't know what the answer was, and they wanted to be able to rank their models themselves. And something that's interesting is that this PLDDT, though it was never intended to be anything but sort of an internal confidence measure, it's turned out it's been demonstrated to correlate with other interesting things like protein disorder. It's actually like the state-of-the-art disorder predictor is AlphaFold PLDDT now. That's a great question. Yeah, because people would like to think that what AlphaFold is giving them is the lowest free energy structure, right? Like that would be nice. But it's not the case. And these are things that are super interesting to me. And it is essentially the second thing you said of just what has it seen so far. And so for some of the systems that I study, I mean, many proteins exist in complexes, and there's often evolutionary pressure put upon something more in its state in a complex rather than if it has an alternative fold that is something else. That might be it's the lowest free energy if it's by itself. And so part of what I mentioned of my work was disentangling this for a particular system where we really know a lot about its different contexts and could say something meaningfully about it. But it's a very interesting question that I think we still don't fully have a picture of. So, so empirically, what we found, again, like for this protein language model, was that it needed about 40 residues of context to make its prediction. I mean, we didn't investigate OmegaFold, but that was the same question that motivated us to investigate the ESM models, is because yeah.
MSA.
You don't have an MSA. No, they don't. Yeah, they don't. But they've learned evolutionary statistics across families. Yeah.
Someone just starting with the protein language model, like, do you have any guidance on like which model you might start with, either practical considerations or theoretical considerations, like, whatever it needs to run out of the box is what you start with?
Yeah. So people talk about flagship models, and there seem to be sort of models that people end up coalescing around. Maybe not necessarily, like they're probably state of the art at some point, but then they also just happen to be sufficiently user friendly and easy enough to hack and ask meaningful questions. And so sort of ESM is one of those for protein language models. AlphaFold is definitely one of those for structure supervised models. I have used Hugging Face, sort of depending on the question and how old the thing is that I want to look at, if it's been moved into Hugging Face or not yet. Did you have your hand up? Right. To me, that is sort of the interesting frontier currently. And so maybe just one way that people are, this isn't quite ionic concentration, but so for instance, Gabe Rocklin at Northwestern is someone who collects massively high throughput stability data. And so his lab is collaborating with the ESM team and others to sort of figure out how can we, because his lab's data still is tiny in comparison to all the sequences, but how could we concurrently use that type of information? Thank you so much.